Hi, everybody, and welcome to this episode of the Combinate Podcast. I'm your host, Subi Sadeh. On this episode, I was joined by my good friend, David Simoens, who is the founder and CEO of Life Science Professionals. During the last time that David and I talked, we talked about aseptic manufacturing in clean rooms. We wanted to also talk about Lyo, but we didn't get to it, and so that's what this episode is. Interestingly enough, we didn't actually get to fully cover Lyo. This episode is specifically focused on the lyophilization process. I learned a ton from David. We cover the freezing process, the primary drying process called sublimation. We cover the secondary drying process, desorption, backfilling, stoppering. He talks about the different considerations for lyophilization, what the impact of cytotoxics, potent or hormone type drugs is on lyophilization, and the different kinds of stoppers used for lyo drugs, differences between lyo and liquid or serum filling vials and processes. And yeah, this is just a really good episode. I learned a lot. I hope you do too. And without further ado, here is David.
Hello, Combi Nation. My name is Subi Sadeh. I've spent over a decade in medical device, pharma, and combination product development. Our industry feels complicated sometimes. Drugs, devices, clinical trials, submissions, sterilization, validation, design control, risk management, market access, reimbursement. The list goes on. My goal is mastery, so this podcast is to ask questions I have to people who may have the answers. Each week on the Combinate Podcast, I talk to someone about their area to further understand and simplify. Whether you're a pharma person trying to understand the next wave of products, or a device person trying to navigate a pharma system you're unfamiliar with, or a newbie in both areas, I invite you to listen, and together we can simplify by combinating.
Lyophilization is an important topic, in my opinion, to understand because a lot of times when we're talking about reconstitution kits and things of the like, the limiting factor is the formulation. So you end up having to build an entire combination product around the fact that you have a lyo drug. And it's important to understand how those drugs are filled because they are different than liquid filled vials. David, maybe let's start with the why. What are the factors that would make one consider lyophilizing versus liquid filling a drug? And is it typically well understood at the immediate in the early stages of a formulation, or is it something that is like, all right, we clearly can't do this in liquid?
I can just give the definition of the lyophilization process and why it's used. Lyophilization is a stabilizing process where a substance is frozen and then a quantity of the solvent is reduced. First, you're using sublimation, which is primary drying, and then you're using desorption, which is secondary drying. And it's to values that will no longer support chemical or biological reactions. So it's stabilizing the drug.
But that's because it wouldn't be stable in liquid.
Right.
When we say solvent, what do we mean?
Solvent,
Because
yeah, solvent is usually,
I'll say my audience is in many cases medical device, and so
Mhm.
they're not as maybe chemically inclined, or maybe they are and I'm just not.
Yeah, solvent is often the portion of the formulation that the API or the active pharmaceutical ingredient is dissolved within. So it's typically WFI, water for injection, but it could also be some form of alcohol or other liquid that it's dissolved within.
And so you said there are two drying steps. One of them, the first one is sublimation, and that's
Yeah,
the
so I
primary
can
drying.
so I can get in like the freeze drying process in a bit. I think you touched on it in the very beginning. There's kind of some part of the process upstream that leads to a successful lyophilization. One of the major differences in vial filling, a typical vial in an aseptic fill finish world goes through either if it's ready to sterilize, so the vials are not currently sterile when they're brought in as raw materials essentially or commodities. They go through a vial washing process followed by a depyrogenation process. and then they're filled aseptically. There's an alternative to that where you can buy ready-to-use commodities, and those vials come pre-sterilized, ready to use, hence the term ready to use. Now, when the vials are being filled, if they're
And
being
in the RTU, there's no vial washing depyrogenation?
typically no, correct.
Okay.
It's usually directly loaded onto a filler, or it's bypassed through a washer in a tunnel, but typically it's loaded directly onto a filler. It's more practical for, say, small batch or for high cost drugs because you're able to mitigate any kind of risk of washer in your tunnel and being able to go directly onto your filler. It's often used too for robotic applications because a lot of RTU ready to use vials can come pre-packed in trays that are able to be more successfully integrated with robotics.
When you say bypass, right, that's for common equipment where there's different types of vials being run through. So bypass means it passes. I've seen it with check weighers and other stuff where there's upstream controls where they're like, we don't need to weigh this, but you're saying it physically passes through the depyrogenation tunnel or the washing station without being washed or depyrogenated.
Yeah, and that's probably more rare. Usually the typical process would be that it's directly loaded on the filler if they're ready to use. So the filling of the vial, if it is a liquid, non-lyophilized product, after the vial is filled, there's a stopper that is placed on the vial, and that serum stopper is placed all the way down into the vial. So the top of the stopper is right on top of the glass versus for a lyo process, after the liquid or the drug product is put into the vial, the stopper is seated halfway into the top of the vial. So what that does, and you're using a specialized lyo stopper, which creates a area where the solvent can escape during the lyo process. So that's a
Um,
key difference in the
are,
filling.
one thing I've always wondered about the lyo stoppers is during that first when it's first applied and it's because it's closed in steps basically. There's a first closure where the it's not sealed fully, like you said, to allow the solvent to escape during freeze drying. But is it considered sealed from a contamination standpoint, or it
No.
still needs to be in a clean room?
Yep, it still needs to be in grade A. Well, there's arguments that it can be in grade A airflow within a grade B or grade C room, as long as you have that grade A airflow, with it still open and not fully capped crimped. So even with a liquid non-lyo fill, the vial is not considered sealed until it's capped and crimped. Even with the stopper fully seated, it's not considered sealed until it's capped and crimped.
Understood. And are there any what unique characteristics are there for a lyo stopper versus a serum stopper?
Yeah, so there's a bunch of differences. So
Is that right?
lyo stoppers can come in many different forms. There's there's an igloo style, which is basically like this. I'll try and draw it for you. So it'll be a nice clip. So it's like this like horseshoe shape, so that when it's seated into the vial, you have like one opening. Then there's other ones that are like two-legged, three-legged. They're referred to stoppers, which have like little rubber legs coming off of the top of the stopper. And so those are two-legged, three-legged. Both igloo style and the three-legged, two-legged stoppers usually have some form of rubber line or indent or something halfway up the stopper so that it is always seated at the same height and can be consistent then across the whole vial shelf stack.
Now, lyo drugs, are they filled under vacuum or are they sealed under vacuum?
Okay, so now we can get into like the rest of the process. So after the vial is filled and the stopper is placed halfway in, then it has to be transferred to the lyophilizer or the freeze dryer.
Is it,
And
are they
so,
not, is it not typically continuous line? It's typically like one line for filling and then it moves?
well, so there's multiple different processes. Sometimes it depends whether it's like a cyto or a potent line, or if it's non-cyto, non-potent, non-hormone type bulk type lines. But there's many different ways. Some are transferred via like a cart or a cart that can either like travel on rails. Sometimes the vials are transferred robotically. Sometimes the vials are transferred in what's called a row by row. So basically, it goes down a conveyor and then it's pushed into the lyo row by row. So it kind of depends on
Oh,
the application. Um, but it can continuous. I think there's a big push in the aseptic sterile fill finish world is like this continuous manufacturing. The thing with freeze dry and lyophilizing is the process takes a long time in the terms of pharma world, right? Some cycles can last up to 3 days, 72 hours. Like cycles can be fairly long. So in addition to like the continuous manufacturing, there's also this big push on, well, how do we reduce lyo cycle times? And I think that's where we can start talking about, well, what is the lyo cycle? So the lyo cycle, it starts out after the vials are in the lyo, and vials need to fill up a whole shelf. Okay, regardless of the size of the freeze dryer, whether it's a 10 square meter freeze dryer or 52 square meter freeze dryer. When you're filling a shelf, the shelf has to be full of vials and typically has to have stoppers on the vast majority of the vials. The reason you want to do this, even if the vial isn't full of drug product, the reason you have to do this is if you don't, the shelves will warp during the stoppering process. So you want to have a full shelf full of vials.
and just to understand the point that you were making. So if you had a small volume Lyo product, you would have to fill the shelves with empty stoppered
Not all the
vials.
shelves. Not all the shelves, but any shelf. So say a shelf will, and I'm going to just like, this isn't realistic, but I'm going to try and simplify it. So say that a shelf will hold 100 vials. Okay. And you fill, and say there's like 5 shelves in the lyo. So each one will hold 100. So the total lyo could hold up to 500 vials.
Yeah.
You only fill 150 vials. So you'd have one full shelf of vials with drug product, and then you'd have a second shelf with half of drug filled product. And that second shelf, you would want to fill it out to make it a full 100 vials. So you'd have 200 vials in the lyo, 150 of them would have drug product, 50 of them would be empty, and 2 full shelves. And so it can get the full compression per shelf, but those other 3 shelves would then be empty.
The and I could be completely wrong when it comes to this, but that second closure of the stopper, it's just it's just a big, there's no vial to, they're not closing each stopper individually, right? That's your point.
No,
They're
no.
closed. It's
So
just one big slab is closing all of them at once.
yeah, and so stoppering's at the end of the process. So like I
Okay.
can go into more in detail here in a second. But like the short answer to be is no. So when the stoppers are fully seated into the vial, is how we refer to it, the shelves come down, usually hydraulics. They push down and push the stoppers into the full shelf of vials.
Okay. Now we talked about the different stopper types. We talked
Hmm.
about the fact that the cycle can last up to 3 days, and just the kind of startup instruction consideration of when you're going through, if a Lyo has a capacity of, let's say, 500 vials over the course of 5 shelves, then you need to make sure that whatever shelves have some utilization are full.
Correct.
Okay. And so let's say you have your 150 vials in, you've added your 50 blanks with
Yes.
the stopper halfway, then what?
Yep. Yeah. So once your vials are in the freeze dryer, that's when the process actually starts. sometimes the shelves will be, and the shelves are what control the freezing aspect. So the first part is freezing. Sometimes they can be pre-chilled so that when the vials are loaded, they're immediately hitting a cold shelf. There's pros and cons to it. Some people claim that there's condensate that can form on the shelves that would lead to vials tipping or those types of things. So some people prefer not a pre-chilled shelf. Some prefer the pre-chilled shelves, especially when you're working with temperature sensitive products that can't be above 2 to 8 degrees Celsius or something. So the freezing stage is the first stage. And basically it's freezing the vial, freezing the products so that you are now in a solid state within the vial. So freezing is accomplished. After freezing, it goes into primary drying, which again is sublimation, which I'll explain here in a second. But basically, it's all around like the triple point of water or triple point of the liquid, where you're in that liquid state, you're freezing it with temperature to go to a solid. Now, in primary drying, you are pulling a vacuum, so you're decreasing the pressure in the chamber, and then slowly raising the temperature so that you're going directly from a solid state to the vapor state, bypassing the liquid state, and that then removes solvent. Secondary drying is then taking that vacuum slightly higher in temperature, slightly higher, or sorry, not taking the vacuum higher, decreasing the vacuum, increasing the pressure, and then raising that temperature so that now you're doing more desorption. And that's like the unfrozen water that's being removed, or unfrozen solvent. It's being pulled off. After freezing, after primary drying, after secondary drying, that's when you go into backfilling the vial with some form of aeration. Often it can be nitrogen, right? Especially if you're trying to reduce oxygen headspace, you can use nitrogen for aeration. So under vacuum, you're putting nitrogen into the chamber, it's going in the vials, and then you're stoppering under a slight vacuum so that nitrogen stays in the vials. And it also helps hold the stopper in the vial with that slight vacuum. And then when you're able to release the vacuum, you can then proceed with opening the doors and unloading.
Okay, so a lot there. Maybe let's
Yes.
start. Okay, so maybe this is something that I hadn't realized in the past, and it seems obvious when people say freeze drying, but I hadn't realized that the drug is frozen solid
Yes.
completely. And how long does it take for that process to occur? I know it's going to
That
differ between drug to drug, but is that the main, or is that just a small part of the process?
Well, so this is where you start getting into some of the science in freeze drying and a lot of what's being discussed in R&D, S&T settings, right? So there are some basic principles that I'm more on, like the
Equipment manufacturing.
equipment design, engineering, project management, delivery side of commercial or bringing in small scale freeze dryers, not so much on like formulation, process development, that type of stuff. But I do understand some of the basic principles.
Yeah. I hadn't realized that it seems obvious that when you're talking about freeze drying, there's a freezing element, but I hadn't realized that it's frozen solid before you move into it. Let's freeze, then dry. And so from that perspective, like what portion of the process is the freezing part time-wise, and what
Yep.
portion is the drying part?
Yeah, and different products and different companies approach it differently. There's an argument. So when you're freezing water, if you're freezing water quickly, it creates small crystals, right? The ice has smaller crystals. If you're freezing something slowly, you have much bigger crystals. Okay. And how does that then impact primary drying? Well, if you have larger crystals, it allows primary drying to go faster. If you have smaller crystals, it usually slows down primary drying. There's some trade-off on, well, if we freeze faster, primary drying might take longer. And if we freeze slower, primary drying will probably go quicker. So that's where like individual product development is important to understand your crystal structures, understand your process and how you're optimizing that, right? How are you fitting it within the design space of the product?
Then the primary drying steps, and you said it's pulling vacuum and raising temperature, and the intent of this step is to go from solid straight into vapor. And how much of the liquid content is removed typically during, obviously it's going to be different, but is it
Is
and
usually
so
the vast majority. Yeah,
like
primary drying
are we talking in the 99% or
probably in the 90% would be safe, right? I think so. Primary drying is, and I don't want to speak for the whole Lyo community, but I'm pretty sure
sure?
primary drying would be considered the main step, right? It's the bulk of what freeze drying is, right? Now, I'm not saying it's any more or less important than freezing. I'm not saying it's more or less important than secondary drying, but it's usually the bulk of the freeze drying process,
Time wise?
time wise, and just like what you're trying to achieve.
And it works by pulling a vacuum and slowly raising temperature? Because
Yes.
you're
So,
and because we talked about, you were talking about how lyophilization is a stabilization process early on,
yes.
right?
Yep.
How do you make sure, like, how high does the temperature go?
So let me take a step back. So freeze dryers, a cycle typically consists of what the industry calls ramps and soaks, right? So basically, it would be a drop in pressure using the vacuum, and then a slow incremental increase in temperature, and then holding at that temperature for a given time,
Understood.
right? Then ramping to a next temperature over a given time at a given rate, and then holding at that temperature. So it's a series of ramping and soaking or holding to do your cycle.
Yeah,
Um,
just like an autoclave cycle.
yes. Yeah.
Yeah. Okay. So what you're saying is they pull the vacuum and then there'll be some change of the temperature till you get to that hold time, and then you have a specific amount of time where you have the vacuum pulled and you've reached the appropriate temperature and you're just sitting there and the
Yeah. I
process is
mean,
working.
you're pulling shelf temperature sometimes negative 40 degrees Celsius. Like it's quite extreme cold,
Cold.
right?
And then when it increases,
Yeah, you're increasing by 10, 15 degrees. Like it really varies by product. And like that's where most of the product development comes in. But these large scale commercial freeze dryers or even smaller ones now for small batch or even for development, you really can have a lot of control around those critical process parameters, right? Though, and by critical process parameters, it's your pressure, your temperature, and your time.
does humidity play a role?
I would say no, because like between the elements of your temperature, your time, and your pressure, like you're controlling how fast you want that vapor to release. There's nothing necessarily measuring the humidity within the freeze dryer as far as like instrumentation or anything. There's a lot more we could get into that would be more like an advanced lyo thing as far as like your temperature collapse point or your product collapse points. What temperature pressure does your cake collapse from being frozen to basically back to a liquid? What is that collapse point? There's a lot of like your basically like your pinch points between your chamber and your condenser and like how fast you could actually send vapor through that, like, and we don't
We
do
can
that here. Yes,
Yeah,
yeah.
we can do that, and this will be like the intro. But I guess what I'm trying to get at is what you were saying about the temperatures. You said you're going from -40, like an extreme deep freeze, or -80 or whatever, and you're going up by 15 degrees, and that's enough to
It could be. Honestly, it really depends on the product and how that cycle is set up.
You're saying it's still pretty much freezing in there, but it's
If you think
drying.
about the triple point, right? If you think about the triple point of water,
What does that mean?
so the triple point is the zero, roughly like it's 0.006 atm of pressure to 0.01 degrees Celsius. So that's the technical triple point of water. And that's when, in theory, water would not be a liquid, it wouldn't be a solid, and it wouldn't be a gas. It's like in between all three, right? And then any fluctuation in temperature or pressure, whether it be up or down, would put it into one of those three states.
Interesting.
So
Okay.
that's really what it's based off of. When you're a solid, so you're ice, and you decrease the pressure by pulling that extreme vacuum, then you can increase the temperature significant. If you're in a deep vacuum and you increase the temperature sometimes to 20 degrees, you're still going to be putting off gas, sublimating, and not be a liquid.
Now, how low do you have to get the pressure? Yeah, because
I
I'm
mean,
looking at it
you
and
can't achieve absolute vacuum or absolute zero as far as a vacuum, but you can get pretty dang close. You can really, with a freeze dryer, you can pull a pretty significant vacuum.
well, that's so interesting. Okay. So
It is.
yeah, yeah. So I'm sorry, I'm just looking at the and so this is physical chemistry, right? Yeah.
Yes.
Didn't have to go that far in college. But this is very interesting that you can maybe I wasn't really aware that ice goes straight from it'll go from and so that's where it says that there's a 0.0006 atmospheric pressure and so it gets that close to if you get ultra close to vacuum right around that 0 to 1 degree mark, you can start to just go
Yeah,
straight from ice
you're
to
not like, it depends on like where in the world you are and what units are using. Millitor is an often used kind of pressure when you're talking about deep vacuum. And you can achieve extremely low vacuum with these systems, or extremely low pressure, excuse me, with these systems to the point where, yes, and that's where I'm saying like the bulk of what lyophilization is that primary drying. It's that, okay, we're frozen in a solid state and we're gonna sublimate off the gas.
now when you go through the primary drying step and you go from ice to vapor, this is probably a stupid question, but no drug product is lost, right? It's just solvent.
Well, and there's a lot of argument around that, especially when you're take like the potent cytotoxic world, where now you have drug that could be dangerous to the operator or the
Right.
facility, right? Like there's a lot of, well, what in the vial is being sublimated or pulled out of the vial, right? What product? And like the key thing I always talk about is, well, what is the molecular weight? Right? The molecular weight is significantly higher. And so it's going to stay for the most part in the bottom of that vial. There are some different cycles when they're very extreme where it'll start to creep up the side of the glass. And like that's where like defects get in and it could go down a whole different conversation. But like usually the
So
starting point.
it would be considered a potential defect if you have cake starting to form on the sides of the vial.
Yes, it typically means that your cycles are too aggressive,
Aggressive. Oh,
right? You like, and again, it depends on the product and like what you consider as still good product quality. But a typical nice looking cake is just in the bottom of the vial and it's not creeping up towards that stopper. Yeah.
interesting. Sublimation is where we go from solid to vapor. Then you move into desorption, which is the catch-all for any remaining moisture that's still in the vial. During sublimation, you pull vacuum and you raise temperature and you hold. What is the cycle like for desorption?
So desorption is usually a slight increase in the temperature. So you're still under that deep vacuum. You're just pushing the temperature significantly higher.
Oh,
Sometimes that's even above, right? You're pushing it above freezing, right? So you're pushing it above 0 degrees potentially, but you're still in that high, deep vacuum.
and again, maybe some of the stuff I hadn't pieced together, but when it's under vacuum, it's basically turning the uh at a molecular level of the drug, it's turning the solvent from a solid to a vapor and literally just sucking it out.
Yeah, and that's where we can get into the equipment eventually, but the vacuum system is usually downstream of the condenser. And so it's pulling the pressure differential between the condenser and the chamber basically pulls all that solvent to the condenser, where it's then condensed and put back to a solid state on some form of coils within the condenser.
Uh-huh. Okay. When you've exited sublimation, most of your moisture content is gone. When you've
Yeah.
exited your desorption, all of your moisture content is gone, theoretically speaking.
And not
Um,
necessarily all, but to the points of what you're trying to achieve, right? So a lot of these
oh,
light cakes might still have a small percentage of moisture in it, but it's that secondary drying that is pulling out those unfrozen water molecules. So the non-ice crystals that's pulled out during sublimation, you're pulling out that unfrozen water in desorption to achieve your moisture content, which would be a critical quality attribute.
and then you move into backfilling. I've heard on the nitrogen side it called nitrogen sparging, at least in the processes that I've worked on. Is nitrogen the only gas that's used? Is a gas always used?
I would say nitrogen is the most common. Sometimes no, if it's not extremely oxygen sensitive, like you can aerate with clean air. But I would say the vast majority of systems and products are now using nitrogen aeration just to, like, if you're trying to stabilize a product, usually oxygen is bad for that, right? So you're using
Oh,
nitrogen to remove what could be more oxygen. And I guess like the basic process is now that you have secondary drying complete and you move into that aeration portion of the process, or backfilling, you would then still be under a small vacuum. You would introduce nitrogen to the chamber, and then you would seat the stoppers all the way so that they're not sealed, but the stoppers are all the way down on the glass on the vial. And then that would allow you to bring the pressure back up to atmosphere so that you can unload. But that holds that small amount of nitrogen in the vial.
and then it moves on into are they ever labeled going into a Lyo, or are they always bright stock?
Typically, no, they're not labeled. Now, I will say there is a big push from a track and trace aggregation standpoint to track each individual vial that's going in based on when it was filled, when it goes in, when it's coming out. There's a lot more technology that's needed to be able to do that for freeze dryers. A lot of even freeze dryers installed in the last 5 years that would be considered new don't have that technology. But no, typically they're never labeled going, and I'll say I have never heard of a case of them being labeled prior to going in. One of the reasons is that would impact the dynamics of the freezing process and how that glass, receives the temperature for freezing.
Well, very good. I think that's the whole process. Did we miss anything
No,
from
I
a
mean, it
process
would
ever?
just move, an unloading, same thing. It could be unloaded row by row with like a push bar or something that's usually at the back of the freeze dryer. So that would hydraulically push out the vials row by row. Or if it's like a T-car system that would mount up to the freeze dryer, put some form of arm in and pull the whole shelf stack out. So it'd unload and then it would go to your cap crimp and downstream process.
The cap crimp is not in the Lyo. And is there any difference between capping and crimping Lyo product, or is it identical?
No, it's usually the same equipment. At the point where the vial is arriving to the cap crimp machine, they would be the same state as if they were a liquid. It's the
Liquid.
stoppers fully seated at that point.
All right, David. Well, we're gonna have to do another episode on the equipment, but where can people find you and tell folks about what do you do?
Yes, people could find me on LinkedIn. I'm fairly active on LinkedIn. I like to do a bunch of posts and stuff within life sciences. So you can find me on LinkedIn. My company page is also on LinkedIn, Life Science Professionals. So you could go there, and website is going to be dropping here soon. So I'm excited about that too.
Awesome. And what kind of work do you do?
So we do consulting within the life science industry, project management, engineering, quality, product launches. We also do a lot with resourcing and staffing, helping find contractors that are life science contractors and consultants within life sciences. So in really all thing life sciences, that's our focus. We don't want to get involved in the gas chemical industry or anything like that. We're just simply focused on life sciences.
Yeah, and David has a real interesting storied start in the food industry, which you can hear in the
I
last
do,
episode
yes.
if you're
Yes.
interested. But thanks for coming on, David. This was awesome. Very good. Thanks, David.
Thanks, Subi.